Alright, so now that we've gone through the abstract intro and discussion and we have a pretty good idea of what the authors claim to have shown, what they think their data in the various figures has demonstrated, we can now start to go through the results. And what I do is I jump back and forth between the figures and the results immediately. I don't just read through the results directly. I want to kind of relate these two together because the results are basically going to give a narrative of their figures along the way. And luckily they pretty much go in order, so if I want to understand figure 1, I'm going to start to read the results and look for key things. I'm going to skip through a lot of this pretty quickly. I'm going to give you a very detailed description of figure 4, but I really want to show you that you can find a lot of these things along the way. So even just in this very first paragraph here, I see a lot of figure 1s, so if I wanted to know exactly what figure 1a is, I'm probably going to read that right up front there. But I can get the take-home message of the first couple parts of it, at least, figure 1a, 1b, right here. These results suggest efficient proteolytic processing of SARS-2S in human cells, in keeping with the presence of several arginine residues at the S1-S2 cleavage site of SARS-2S, but not SARS-1. In contrast, this S2 cleavage site was similar in both. So what are they talking about? Well, now if I go back here, I can see that they're comparing the spike protein, that protein that binds to the receptor and allows the virus to get in. They're comparing that sequence between the original SARS and SARS-2, and they're comparing it at two sites, S1, S2, and S2'. And you can see that at S2', they're very similar, and they already know that the original SARS gets cleaved there. They also know that some other coronaviruses can actually have a cleavage at this site too. Not the original SARS, but some other ones. And here we see that this is a pretty big difference between the two. So that might be a key difference. And then they go on, and I saw this in the results as well. I'm going to skip through it here. But they go on to show that the spike protein indeed does get cleaved at two sites. All right? So let's kind of move on from there to some of the other parts that are a little bit more important. So what about figure 1c? Well, we can kind of go through this, and then they'll basically tell you what their next question was. We first asked which cell lines were susceptible to the SARS-2S driven entry, using a panel of well-characterized cell lines of human and animal origin respectively. So they took a lot of cell lines where they know what those cells express, and they basically decided to try and figure out which ones are susceptible to infection by COVID-19, SARS-S2. All right, and so we can see that up here. This is what they're doing. These are all the various cell lines. And these cell lines here in the gray are human cell lines. Then you have one from monkey here. The Vero cell line is from monkey. And then we have pig, cow. We have all these various cell lines. The VSVG, they say in their results as well, and I can look that up. This is basically a positive control virus. This is a shell that infects all cells. All right, and so they're using that really to get to that. So where did I see that? All right. So all cell lines were readily susceptible to entry driven by the glycoprotein of the pantropic VSV. So pantropic, you might have to look that up, but pantropic basically means that this is something that has a tropic nature to everything. It can infect everything, pantropic. So as expected. Basically they're using that as their positive cell line, and they just to make sure that everything's working right. And indeed, every cell line is infected by this virus, pretty high levels. But when they went to SARS-2 and SARS-2S, so again we can kind of go back down here, most human cell lines and the animal cell lines Vero, that's the monkey cell line, and MDCK2 were also susceptible by SARS-S, and SARS-2S had an identical spectrum of cell lines as SARS-S. So what they're showing here is that any cell line that was infected by SARS-S, that original SARS from a couple decades ago, was also infected in a very similar pattern by SARS-2, the COVID-19 SARS. And this includes most of the human cell lines, that monkey cell line, and this MDCK2. And then they go on to make that speculation, and this is key when you're presenting this and also when you're understanding an article. You want to know the take-home message. So the take-home message so far here is that the spike protein gets processed, All right, so they're going to go on and further and trying to figure out how it's primed, how it's processed. And the SARS-CoV-19 virus has a similar infection to one of the previous SARS viruses, SARS-S. And they then say, well, this leads us to believe that it's possible that the receptor for SARS-2S is the same as the receptors for SARS-S. And that is a key fact because they already know the receptor for SARS-S. And if SARS-2S is infecting the exact same set of cells, it's a good possibility that it's using the same receptor because otherwise there would be different sets of cells. There might be some overlap, right? So these cells might express several of the same membrane proteins as some other cells. However, chances are good that it wouldn't be the exact same set of cells. SARS-2S would infect some cells that the original didn't and would not infect some that it did. The fact that they infect the exact same cells leads them to believe that might be the same receptor. And this then starts to clue us into figure 2. And so if we go to figure 2, in order to elucidate why they enter the same cell lines, we determined whether SARS-2S has the same amino acid residues required for interaction with the original SARS entry receptor. So basically, they're now going to compare those two spike proteins, especially the areas that they know is important for SARS-S using the receptor ACE2 and see if there's similarities. So that's a transition. From figure 1, they now know that this new SARS virus infects the same cells as one of the original ones. So they now want to find out if the spike protein is similar at key spots that allowed the original one to bind to ACE2. Could ACE2 also be the receptor for the new one? So that leads to figure 2. I'm going to skip through this quite a bit, but this is that analysis. And there are key proteins that they already know are important for binding to ACE2. And you can see those here with SARS and a few other viruses that use ACE2 as the receptor. And you can see that SARS-2 lines up perfectly, or nearly perfectly with those. And not such similarity with coronaviruses that do not bind to ACE2. So these are all coronaviruses that bind ACE2 as a receptor. SARS seems to fit that profile. So you can probably imagine what they're going to do next in figure 3. Well, that means in figure 3 they're going to see if SARS-CoV-2 is using that. So let's jump to figure 3. Another thing you can use to really determine what the figure is showing is the figure legend. So between the figure legend and those results sections, you can have a pretty good idea of what the figure is showing, especially when we have those conclusions from the discussion in the back of our mind. We already knew that they'd shown that there was priming, and they'd shown that COVID-19 virus uses the same ACE2 receptor as original SARS. Now we're seeing how they showed that, how they figured that out. So let's see what they're doing here. BHK21 cells, right? So they just used cell line, transiently expressing ACE2. So they make this cell line express ACE2, right? And so this is important. This is a little bit subtle, and you might not have gotten it, but this cell line, which was, sorry, I forgot already which one it was. I know I'm making you dizzy, sorry. BHK21. Those BHK21 cells, right here, BHK21 cells were not infected by the virus because those BHK21 cells don't express ACE2. So if you make those BHK21 cells express ACE2 and now suddenly the virus enters, that's a pretty good indication that ACE2 must be the receptor, because it could not infect those cells until you made those cells express ACE22. So, BHK21 cells expressing ACE2, right, were inoculated and given this virus that has the original SARS, COVID-19, MERS, or 229ES, so some other kind of coronaviruses. So let's see what happened. All right, and so one of the things I meant to point out, I'm sorry that I haven't been mentioning it along the way, Anytime you get one of those take-home messages, anytime you've figured out exactly what a figure is showing, write it down. Because you're going to come back to this and you're going to have thought about a lot of other things along the way. And now I can go right back, okay, what are they doing? These are the positive controls. I might even have more notes here that just say tested infection levels of all the different cell types. And here's the main conclusion. That should be written pretty much on every one of your figures so that you're ready to understand, come back to it, and eventually ready to present. All right? All right, so with that tangent, let's go back to where we were. Sorry. All right, so we know this is our positive control. All right, the positive control can infect anything. This is the virus, or sorry, this is the cell line expressing human ACE2. This is the cell line expressing a bat ACE2. This is the cell line expressing two different receptors, not ACE2. So the positive control infects all of them. The original SARS only infects it if they have the human or the bat ACE2. So in other words, they're not using these receptors. These were other potential receptors that the authors are checking. They hypothesize it's ACE2, but these are other ones that other coronaviruses infect. SARS-2, our COVID-19, same thing. Only infects if it has ACE2, human or bat. And other viruses use the different receptors. Our SARS-2, therefore, is using the same receptor as SARS. And we now know that it can infect these cells if they're expressing ACE2, but not if they're expressing other options. But if you want to really prove that it's ACE2, you now block that ACE2 again. All right, so we come down here. Untreated viro cells. All right, so why viro cells? Well, we learned why they were using the BHK21 cells because those couldn't be infected, and so by making them express ACE2, we were showing that now when the virus can infect it, ACE2 must have been important. Right? But viro cells, those could be infected. Right? Those were those monkey cells that were infected already. So now we want to see if that infection was because they were expressing ACE2. So if we block ACE2, will we no longer get infection? And our positive control works all the way through. Basically, the original MERS virus that wasn't using ACE2, when you block ACE2 with this antibody, there's no effect. But when you start blocking this with the ACE2 antibody, now the original SARS and COVID-19 both stop getting in. So now you've really proven that ACE2 is the receptor because cells that it could not infect, it is able to infect when you express ACE2. And cells that it could infect before, it no longer can infect when you block ACE2. These are the types of things you're looking for. And again, I would have my take-home message written all over here. When you're presenting, keep those things in mind. And these are key transition statements. Now that they know this, they then wanted to find out this. And they were trying to find this out by, so in this case, they were trying to find out if ACE2 was indeed the receptor by taking cells that COVID-19 did not infect, making them express ACE2, and now they see that they did infect it. Then they took cells that COVID-19 could infect, blocked ACE2, and determined that it couldn't infect it anymore. These are the take-home messages that you want to understand and you want to present to the audience when you're getting there. All right, I am next going to stop this video, and I am going to give a full kind of presentation walking you through Figure 4, through one of the complex ones, and then you're going to analyze Figure 5 on your own and see if you can figure out what that is, and that will basically be the extent of this activity. And then I will have one more video giving you hints on how to present. I've had a lot through here, so hopefully you've taken some ideas along the way from here with those transition statements, those take-home messages, and that aspect. But I'll have that last video, and then that's it for this lab. All right.